I am going to create the multiple copies of the same gene. That is the recombinant gene therapy what we have done so far. So stem cell is the unique cell which is undifferentiated, keeps multiplying and differentiated into organs. Like example, like here. This first is embryonic stem cell. Fine. Listen. This is the embryonic stem cell. You see here. This is the first stem cell. It's the first stem cell. This is embryonic stem cell. This embryonic stem cell is pluripotent, means multiple possibilities. And the source of this embryonic stem cell is what? It's a 3 to 5 days old embryo. It's a 3 to 5 days old embryo. Like see, it's a fertilized egg. This is zygote and it starts multiplying. This is what? It's an embryo. This is embryo. It's a 3 to 5 days old embryo. It's a 3 to 5 days old embryo, and we all develop like this. So if you just look at this structure here, there are so many cells inside like this. These all cells are what? Undifferentiated. All cells are undifferentiated. Yes? But these undifferentiated cells are going to be differentiated in the different body parts. So in the process of differentiation, it is going to be developed specialized cells of the body, like the heart cells, liver cells, pancreatic cells, intestinal cells, blood cells. Means the entire body parts are the result of differentiation of these undifferentiated embryonic cells. Yes, that is the natural development we are talking about. Suppose if I am going to isolate few cells, I am going to isolate probably if this region is not visible, so I am just mentioning here. If I am going to isolate few cells from here, this embryo. Fine. So this is what? It's a embryonic stem cell, and this embryonic stem cell is followed by differentiation process. Yes, and then as a result of differentiation, these cells will followed by medication in the patient, whether this medication is the heart problem, whether kidney problem, whether liver problem, whether pancreatic problem, or X, Y, Z. So suppose if we have isolated the embryonic stem cell and then we are going to separate it. So as per the requirement, we are going to inject in the body, and that is going to heal patient. The entire body is what? The entire body is the cellular, and if any body part is not functioning properly, in that case we are supposed to medicate that particular body part with the help of a cell, right? And the source of that cell is what? It's an embryonic stem cell, because embryo has the ability to develop all body parts, right? So embryo cell has ability to develop most of the body part that make a sense of the pluripotency. So the embryonic stem cell is pluripotent in nature, what we have mentioned here. Is it clear? But the point is that in the process of the extraction of embryonic stem cell, probably we are going to compromise with the embryos. Means once we are going to isolate a stem cell from here, which is used for medication. Yes, so this will continue, but this embryo is the precursor of next generation. Yes, so in the process of extraction, if I'm going to isolate it, so this next generation will stop, and this is the case of embryonic killing. So for the medication, embryonic killing is not permitted. So the use of embryonic stem cell is what? It is not permitted due to ethical reason. Yes, why? Because if you are going to use this embryonic stem cell, so the result is what? Well, we are going to compromise with the life of the next generation, which is not permitted. Yes, right? See, listen. See, there is a question scrolling on the screen. See, how can it be determined that, listen, what happened? Cell has the gene sequences. Every cell has gene sequences. Yes, every cell has gene sequences. And it is the action of the gene. Is going to determine the nature of the body parts. Fine. So across the body part, we have a similar gene sequences. Across the body part. Yes. But listen what happened. Hair cell is different from the skin cell. Skin cell is different from the heart cell. It is because of the specific gene expression in the specific cell. So every cell has 20 to 25,000 of the gene sequences. But all gene sequences are not expressible in all cells, right? A particular cell is going to express particular gene sequence, and the rest of the gene sequences are non-expressible in that particular cell. So this leads to differentiation in the action of the gene, and as a result of the differentiation in the action of the gene, the different body parts are going to be developed. This is the same way how this development is going to be done. Listen, we all develop like the same. Ultimately, what happened? Now we all resulted in the form of zygote. And then this zygote went through the 9 months of the development, and automatically what happened? The few cells are going to be immersed like the heart, the few cells are going to be immersed like the body part like the hand, leg, the few cells are going to be developed like the brain, the few cells are going to be developed like the hair cells, the few cells are going to be developed into the internal body parts like the intestine, stomach and all and all. So this is because of the differential expression of the gene in a particular cell that leads to differentiation of the body parts. So when we are going to culture these cells, so then after culturing, we are going to instruct that particular cell. Because that cell has all gene sequences, right? That cell has all gene sequences. And we are going to instruct this particular cell means we are going to instruct the gene of a particular gene of a cell to express in a particular body part. That is what? This direction we are providing. The direction we are providing externally. Right? That direction we are providing externally in the lab through the gene signaling process. And it is not like that every time whenever we are going to instruct, the gene or the cell is going to be act like that. In most of the cases, the cell or the gene deviates from the normal function. So the chance of the success rate is what? It is less than 1% because every time it is going to be succeeded is not like that. So it's a very difficult, it's a very technical process what we are talking about. Right? Fine. This is what it's the embryonic stem cell what we have discussed. So why we are talking about even the embryonic stem cell is not permitted. So how we are using this stem cell? How we are using this stem cell in medication? So that is the second example of the stem cell. And the second example of the stem cell is what? It's a adult stem cell. It's a tissue or organ specific stem cell. It's a tissue or organ specific stem cell, and the intention of embryonic stem cell is not permitted. Embryonic stem cell is not permitted for the medication abhyas. What is permitted is the adult stem cell. This adult stem cell is permitted. For medication. And this is called as what? It's a therapeutic clothing. Or this is called as cell-based therapy. So this is what? It's a tissue or organ specific stem cell. And the function is what? Function is repairing and regeneration. And every body part has adult stem cell. Every body part has adult stem cell. Right? Let's see example. The hair is, you are going to cut for the hair. You are going for the haircut, and then what happened? Maybe after a couple of days you are going to see the longer hair. How it is possible? Because at the base of the hair cell there is a stem cell, and the hair stem cell is responsible for the elongation of the hair. Like the blood. The original blood is replaced by the new blood in the next 3 months because in bone marrow there is a stem cell. The skin is replaced, the old skin is replaced by the new skin because there is a stem cell. Means every body part has a stem cell. So adult stem, where exactly the adult stem cell is found? Adult stem cell is found everywhere. It's an organ specific stem cell, umbilical cord, like you heard about the cord, blood, spinal cord, liver, fat, bone marrow, dental pulp, pancreas, brain, peripheral blood, retina, skeletal muscles, skin. So every body part has stem cell. Yes, and the function is what? Repairing and the regeneration of repairing and regeneration. Fine. So the most difficult point is that technical issue is how to isolate. Technical issue is how to isolate. And then after isolation, transplant. Yes? Like example, liver transplantation. You must be knowing about in the case of liver transplantation, we need to take a cut part of the liver from the donors and then transferring to the recipient. That is what? It's a stem cell transplantation. So adult stem cell is what? It is organ specific stem cell. Adult stem cell is organ specific stem cell. Organ specific stem cell, and the intention is we need to take it from the body of the donor and then we are supposed to transfer to the body of the acceptor. So suppose if I am a bone marrow donor, So you take a part of the bone marrow from my body and then get it transferred to the body of the recipient. Clear? And this bone marrow will multiply itself and it is going to result into blood. And that blood is what? That blood is required for the compulsion in living organism is what? It's a bone marrow transplantation. Like the liver, in the case of the liver, only a part of the liver is required from the donor. So we need to take a part of the liver from the donor and then suppose we transfer to the recipient for the liver development. So the entire liver is not been transplanted. So we are heading toward the technology where we are supposed to develop the entire body parts. We are supposed to develop entire body parts. Right? This the body part development would be possible on the basis of the cell-based therapy. On the basis of the cell-based therapy, and if we are going to be succeeded like this, so we are not supposed to be bank upon. We are not supposed to be bank upon the organ requirement from the donor side. Clear? Fine. But the technical point regarding this entire discussion is what? How to isolate and how to transplant is the most crucial aspect of this particular technology. So someone asked about the hair transplantation. Yes, obviously you heard about the permanent solution of the hair transplant. So in that case, this hair is the dead part, and the pulp of the hair is transplanted to the bud. So the pulp of the hair is the good source of the stem cell. The pulp of the hair is the good source of the stem cell, and once we have transplanted the stem cell of the hair, so that stem cell will try to multiply and it will make a new hair, that is the permanent solution of the baldness. Like the same bone marrow transplantation we are doing, like the same liver transplantation we are doing. Fine, so that could be possible. But the point is that where exactly the location of a stem cell in the corresponding body part, that is hard to find out. That's why we are restricted about this technology. Otherwise, probably in the couple of the years, maybe a couple of the decades, we are heading toward the technology, and we are heading toward the technology where we must have a body shop, and that body shop means if any body part is not working, in that case we need to go to the body shop and ask for the replacement of the particular body part. And that particular body part will be the result of the organ cloning or the body part cloning on the basis of this adult stem cell technology we are talking about. Right? Fine? Okay? So this technology is permitted. This technology is permitted, and this adult stem cell is usually it's a unipotent. Means organ specific means what? The liver will always make liver. That is the one-to-one relationship. Skin will always make skin. That is the single possibility. Single possibility. Is the part of liver transplantation same as the tail of the lizard? Yes, the tail of lizard is naturally growing because of the stem cell of tail. Means we are simply extracting the tail of a lizard. But even somewhere the stem cell part is there that starts multiplying, and the result is the new tail. It's like replacing the defected part of the robot. How we are going? We can say like that is that we can consider. We could consider. See here, this is what it's a stem cell possibility. In the body, we do have a provision of more than 200 types of stem cell like this. And this stem cell, this could be used for the growth of the brain, liver, lungs, heart, genitalia, kidney. So why we cannot regrow the lost limb, which is with the help of stem cell technology? We don't have this technology, but it is not like that in near future we are not going to do like the same. We probably in near future that could be possible, but as of now we don't have technology to stimulate the stem cell in the lost part of the limb, because the stem cell also needs stimulation. Right? So in certain areas stem cells are naturally stimulated, like see the bone is broken down. We are not supposed to do like this. Right? The broken bone part of the bone will already been corrected just because of the multiplication of the bone cells. This is a natural regeneration. So in few cases of the stem cells are blessed with the natural tendency for the regrow and the repair, but there are few examples of the stem cell where the stimulation is required. So in the case of limb stem cell, stimulation is required. So could be possible in near future. But as of now, we don't know where exactly the stem cell of limb that we have lost in accident. So if it is possible in the near future, we are supposed to stimulate the stem cell of the limb, and that is after stimulation, this will obviously. The injury healing is done for the stem cell. The injury healing is done for the stem cell. After operation. The recovery is what? The recovery means healing after operation is what? Because of the stem cell. Bone is broken down, the broken part of the bone is healed. That is because of the stem cell. Right? Okay? You know how it is being done? See here. This is the way we are doing. We need to get this stem cell from the donor. Harvest. And we need to take it outside. Then we need to separate corresponding stem cell. The stem cell are separated from the fat cells. We need to separate a specific stem cell. Then we need to activate stem cell. Activation means activation of a specific gene. A specific gene for a specific body part. A specific gene for a specific body part. Like see, this time we need to isolate, we need to express the blood gene. So this specific gene, blood gene is now activated, and then we need to return in body of recipient. This is the way we are doing. Done? Right. So listen, in this man, blood gene is inactive. So we need to replace this blood gene. Anna? We need to replace this original blood gene by new blood gene, like this. And the source of new blood gene is what? It's a bone marrow. That is cell based. And the source of this bone marrow is what? Donor. So we have replaced this faulty gene by healthy gene, and this healthy gene is inserted with the cell. So this is called as what? It's a cell based therapy. Yes, right? Done? Can we go ahead? The next is iPSC. Are you getting transmission properly? Please let me know. Are you getting transmission properly? Induced pluripotent stem cell. This is the new concept. How do we determine who should donate the stem cell? Diksha, this is on the basis of the genetic similarity between the donor and acceptor. And see, in the novel case of the heart transplantation, kidney transplantation, we are just trying to match the blood group and even the genetic matching. So that depends, which ensures the genetic similarity to some extent between the donor and acceptor. Like the same, we are also supposed to match the genetic similarity to the minimum extent between the donor and acceptor. So that will ensure by doctor. If I am going to donate my bone marrow to anyone, so it is not like that the doctor is going to take my bone marrow and it is transferring to the recipient. No, doctor will try to analyze whether my bone marrow is compatible for the recipient bone marrow or not. So doctor will try to maintain the genetic similarity between the donors and the acceptor. Yes. And if the gene sequences to some extent are going to be matched, Right. Then after that, this is going to be this transplantation, this bone marrow transplantation is going to be done. So the induced pluripotent stem cell is a new concept. And here what happened? Adult stem cell, which is unipotent, is reprogrammed at genetic level, and this will leads to many possibilities of cells. So one possibility of a stem cell will turn into the many possibilities of the stem cell, and this is pluripotency, and this is possible by reprogramming. It is possible by reprogramming of gene. So this reprogramming of the gene was first conducted by Yamanaka, Shinya Yamanaka in 2007, and for the same he awarded Nobel Prize in 2012. Fine. So like human, you take my skin cell. So usually this skin cell will develop skin cell, always develop skin cell. But we have simply modified the gene sequences. We have simply reprogrammed the gene sequences we have simply reprogrammed the gene sequences of the skin cell to that extent, so that the skin cell is going to develop, is going to result even the heart cells, kidney cells, because what we have done, because we have simply reprogrammed the other gene sequences apart from the gene which is responsible for the skin. So we have simply created many possibilities, means we have simply induced pluripotency. And that is the logic behind we named it as a iPSC, induced pluripotent stem cell. So this is a new concept, and this is the it's a lab induced concept. And this lab induced concept is, first of all, this concept was developed by Shinya Yamanaka in 2007. Right, and for this revolutionary concept, he awarded Nobel Prize. So definitely, this process, this concept is going to create a great change, especially in the medical science, because the use of embryonic stem cell, which is pluripotent stem cell, is banned. But if you're going to induce pluripotency like this, right? So this is what it is not going to create any ethical problem at any extent. So definitely this is what it's a revolutionary aspect. Okay, and nowadays you heard about lab grown meat. Yes, this lab grown meat it's a synthetic meat. Which is permitted in Singapore. Yes, and this is nothing but it's a adult stem cell. It's an adult stem cell. right? You take a stem cell. Suppose if you love chicken leg piece, है ना? So no need to slaughter chicken. No need to slaughter chicken. Rather, you take the stem cell of chicken leg and get it culture in lab, and you are going to culture leg piece in a lab. So that is what, it's a lab grown meat. So this lab grown meat is the classical example of the stem cell, and it is quite ethical because it does not allow the slaughtering of animals or the birds, and even the quality of this meat is always better as compared to slaughtered meat. Why? Because this is pathogen free meat. It's a disease free meat because we have grown it in the lab. We have grown this meat in lab, right? So definitely this quality of the meat is better than the market means, the slaughtered meat, because there could be the chance of the contamination, but this contamination is least, or this contamination is negligible in the case of the lab grown meat is the concept of the stem cell technology, right? And the question asked by Ritik Sharma, what is meant when a doctor asks about the stem cell conservation at the time of the birth of the baby, which already I mentioned here. See, this is nothing but, Ritik, your answer, this is nothing but, it's a storage of, umbilical cord. It's a cord blood. So that this cord blood is, you have seen the umbilical cord, that is the placental connection between the mother uterus and the baby, which is connected with the navel part of the baby, right? That is the supply line between the baby and the mother. So when we are going to cut, that part is the umbilical cord. When we are going to cut, so it will start bleeding. Clear? So then we need to collect the blood from this bleeded part, and that consists of the stem cell of developing baby, so that the baby is going to be suffer from the developmental irregularities in near future. So in that case, we need to isolate the corresponding stem cell from the cord blood cell what we have collected, right? And that cord blood, that specific cord blood will be used for the medication of the baby, is the logic behind why doctor is trying to convince couple for the conservation of the cord blood, because this is a good way to handle developmental irregularities in developing baby, which is permanent solution. Clear? And even it's nowadays it's a very cheaper, the storage is quite cheaper nowadays, just because of the availability of a number of cord blood bank. What about the protein content of the animal flesh? Obviously the protein content would be, right, the protein content would be better. Means this is the animal grown meat. The animal grown meat is quite proteinaceous in nature, because the slaughtered meat is also source of fat. Fatty part is also there in the animal meat. So that fatty part we are not supposed to culture. Means this as a whole we can say like this animal grown meat is the part of protein. This is what, it's a good source of the protein, and the other part we are simply avoiding when we are growing this meat in the lab. Okay, right? So this is all about therapeutic cloning regarding the stem cell technology that we have done so far. Yes? Okay? And the third cloning is the reproductive cloning. This we are going to discuss in the next class. This is reproductive cloning, or the same is also called as organism cloning. Yes, so this we are going to discuss in tomorrow. Tomorrow means not tomorrow, it's a Monday. The next class